Okay. All right. I'm Brandy Keel-Reese, the PI of this grant. And as I've mentioned, Ruth Carmichael and Sinead Nehine are also co-PIs on the grant. Each of us have a distinct role in in this project. Our students are Anya Brown, Penny Dimitriades, and Bree Jansen. So online, I'm just going to go down the list and who I see. I see Carlotta Davis is here. Can you introduce yourself?
Yes,
Mhm.
yes.
Mhm.
This Carlotta Davis, We Matter Community Association.
Fantastic. Thank you for being here. I see Kevin,
Yes, Kevin Calzi here, Food and Drug.
and next I see Mimi.
I'm Mimi Fern, Dog River Clearwater Revival.
Thank you for being here. I see just Mobile Bay National Estuary Program. Who's online?
Becca Farmer, Restoration Project Coordinator.
Fantastic. I see Kristen Butler.
Yeah, hey, this is Kristen Butler, Senior Microbiologist at FTA.
Great. Thanks for coming. Amanda Free.
Hi, yeah, I'm with Mississippi State University.
Great, thank you. Margot.
Hi, Margot Post. I'm with the Grand Bay National Estuarine Research Reserve over in Moss Point.
Fantastic, thank you. Scott Phipps.
This is Scott. I'm at the Weeks Bay National Estuarine Research Reserve.
Fantastic. I see Shamika Brown.
Hi, Shamika Brown, Mobile Bay National Estuary Community Engagement Program Lead.
Excellent, thank you for being here. I see Matt. Oh, okay, we can come back to Matt. Ted Henkin. Okay. I also see Fred Leslie.
Fred Leslie with the Alabama Department of Environmental Management in Montgomery.
Fantastic, thanks for being here. I see Yu Ching Feng.
Oh, hi. I'm with Auburn University. I'm an environmental microbiologist here.
Thanks for joining. I also see Cassie Bates.
Hi, I'm Patsy Bates. I'm the staff scientist at Mobile Bay Keeper.
Great, thanks for joining. I will come back to Ted Henkin.
Can you hear me now?
Yes, thank you.
Okay, I'm with the Fowl River Community Association.
Fantastic, thanks for joining. And Matt as well. Okay, and that's all right. Maybe just listening in is great as well. All right, so we'll go ahead and get started with, we did our introductions and met our research team. And I'm going to turn it over to Ruth to give the project updates, and then we'll get started with each of our well, actually, here are our community partners, and thank everyone for being here. If you don't see your name listed, please let me know. It was an unintentional oversight. So if you don't see your name or your logo listed, I'd be glad to fix that and to correct that. And I'm going to turn it over to Ruth to introduce more of the project, the objectives, and then we'll hear from each of our students about the updates and their particular research areas.
Good afternoon, everyone. I hope you can hear me okay. I'm kind of shouting, so I hope that works.
That'll be fine. Thank you.
Everybody in my room is hunkering down. I'm going to take cover. Thank you all so much for being here with us this afternoon. And I did want to just do a quick shout out Matt, if you are out there and you want to introduce yourself, you can always post it in the chat, and we'll be able to read whatever you posted if you don't have the ability to use your microphone. So for everyone else, just a quick reminder, I know some of you folks maybe have not been with us before, so we just want to make sure to go through our objectives and remind everyone that our major objectives are really fourfold. First, to define the microbial sources to Alabama waters, with that specific goal to be able to distinguish at least human wastewater, so wastewater treatment plant or septic inputs, from, for example, wildlife like waterfowl, wild boar, livestock, and other types of non-human inputs to our waters. Then to define the specific conditions that, difficult to hear still? All right, I'll try to speak up. To define the conditions that locally, are affecting microbial concentrations and the timing of when we're seeing those in our waters. Also, to facilitate data sharing and using our existing, we have an existing project called Wastewater Footprint, which is essentially an online clearinghouse or a one-stop shop for information on water quality. And we invite you all to visit that website. We are regularly posting information there, sort of updating it, but we have all of the information about this project and our prior projects there as well. And you can get recordings of this presentation as well. So you'll be able to listen to those recordings. So we encourage you to use that platform, which is our third objective, to add to that platform. And then also, finally, to enhance local capacity for long-term tracing and understanding of our microbial inputs through some specific outreach and community activities that we'll talk about in more detail. So at this point, we're going to turn it over to our boots on the ground, who are students who are really making this project happen. And this is Anya Brown, who is working on our traditional microbial indicators and also some of the other indicators of human wastewater, and Penny Dimitriades. And we'll start with Anya.
Hi, everyone. So as many of you may remember, or as a reminder, we started our monthly water sampling back in September 2022. And since our last call, I'm pleased to say that we have finished as of September 2023. And in total, we have collected 14 months worth of water samples. Along the way, I've been doing the processing and analysis for the microbial indicators. So that's the fecal coliforms, the E. coli, and the male-specific coliphage. I've also been processing and analyzing samples for nutrients, stable isotopes, and those are mostly finished. Penny also has been working on source tracking qPCR work, including primer validation, qPCR optimization, and DNA extraction, but she can talk more about that in a bit. But a great thing about this project and our collaborations is that it's allowed us to leverage the time and efforts we're already putting into the water sampling, but add additional components to gather more data and get a more comprehensive picture of water quality. So, for example, I'm also developing assays for direct pathogen testing. That's for Toxoplasma gondii and Erysipelathrix russipathiae, and those are two pathogens of interest to water quality. So since we already have the samples, I'll be analyzing them for the pathogens as well. We are also going to be analyzing the samples for trace elements, and that's including total mercury concentrations. And also, while we've been out there, we've been deploying oysters and collecting sediment samples. We've been doing this seasonally at a subset of our sites, and this is in order to study additional aspects of this system and how it may be impacted by freshwater-related water quality changes. So we've been doing a lot of additional work around just the water sampling and the traditional and novel indicators. You can go to the next slide. So now that sample collection is complete, here are a breakdown of my sample numbers. So as I mentioned, we finished with the microbial indicator samples. So those were processed in-house at our Dauphin Island Sea Lab and FDA joint lab at the University of South Alabama campus. This amounted to a total of 182 samples. So that's 13 sites over 14 months. The stable isotopes have all been processed, and they've all been sent out to the UC Davis Stable Isotope Facility, and I have about half of that data back already. Similarly, I've processed and sent out all of the samples for trace elements, and those were sent to the University of Texas at Austin, the ICP-MS facility there, and I have all of that data back as well. Going forward, I still need to finish some of the nutrient analyses. We're doing nitrate, nitrite, ammonium, and total dissolved nitrogen. So I've processed 11 months of those samples, so I have 3 months left to finish. And those are being processed in-house at the Dauphin Island Sea Lab and the University of South Alabama labs. Similarly, I'm going to add total mercury. That's a separate analysis from the trace elements. So currently that's on hold until we get our equipment up and running and protocols sorted out. But those are the next steps, as well as I mentioned, getting the PCR assays up and running, which is currently in progress, as well as the data processing and analysis. So I've now compiled the complete set of data for the microbial indicators. There's still some more processing to do, but for now, here you can see those indicators by site over time. And as a reminder, there's a map on the left there, and it's color-coded with the sites to the graphs on the right there. And you can see we have fecal coliform numbers on the top right, and E. coli numbers below that, again, over time, monthly, and both are measured in colony forming units per 100 milliliters, and those numbers have been log transformed. You can see a pattern emerges over the year with an increase in the colony forming units, starting around December and increasing over the winter months up until about March, before they start decreasing again. And this pattern matches sort of what we've expected and corresponds to lower salinity waters seen in the winter months. The male-specific coliphage on the next slide. There we go. Shows a similar pattern. So this is in plaque forming units per 100 mil here, and again by site over time. So we're seeing far less of the coliphage overall, but there's still a distinct uptick in the numbers over the winter months, or rather an increase in number of the different sites where we're seeing the MSC pop up. The orange bar is consistently high. That is our wastewater treatment plant site. So we're sampling the effluent there, and that MSC presence is consistent and consistently pretty high compared to any of our other sites.
Anya, before you,
Yes.
Can you remind everyone what MSC is?
So, sorry, the male-specific coliphage, that is our viral indicator. So that's a proxy for like human enteric viruses. So a good indication of human wastewater.
Thank you.
Yeah. So just another way to look at this data is I plotted the microbial indicators versus salinity, so you can see some of these trends. So again, fecal coliforms on top, E. coli on the bottom right there. And you can see this general negative linear relationship. As the salinity decreases, colony counts increase. And then there's a similar pattern with the male-specific coliphage, again, only in lower concentrations. Another way I plotted this data to look at these sites where we're seeing these increased indicator concentrations is to look at over which months. So here I plotted the indicators by site over time. And the key is on the left there. I know they're a little bit small, but I wanted just to like point out some of the general patterns that we're seeing. So the darker lines are in the colder fall and winter months, whereas the lighter colors are in spring and summer. So interestingly, I think you start to see consistently higher contributions from the sites north and east of the bay. So it'll be the right-hand side of each of those sites, as compared to the more southwest sites, which would be on the left-hand side of that. So the Mobile River are pretty consistently high as compared to some of the other sites. Again, this is just male-specific coliphage plotted in the same way, and so a similar pattern emerges, only you really see how consistently MSC was present at the wastewater treatment plant there as compared to any of the other sites. So lastly, I have most of my nutrient data analyzed and processed, as I mentioned, barring the last 3 months. So I just wanted to give a peek at some of the preliminary data here. So this is plotting the concentrations of nitrate, nitrite, and ammonium in parts per million by site over time. So I know it's a little hard to see, it's a little squished in there because there were a lot of sites and a lot of months. But each site is plotted for each month going from September 2022 all the way on the left x-axis to June B, which was our second sampling in June, and that's all the way on the right x-axis down there. And so this is including our wastewater treatment facility site, and it's consistently high in ammonium here. So it kind of skews the data and you can't really see everything else really clearly. And ammonium is sometimes an indicator of raw wastewater presence. So we're still going to look into this and discuss this a little bit more. But to better see the patterns of the other sites, I took out that wastewater treatment facility site. So you can see here higher concentrations of nitrate that, again, increase over the winter months. So that same pattern is emerging, and it decreases again in the spring and summer months. So again, I'm still processing this data and waiting for the rest of the samples, but you can start to see some of those spatial patterns emerging. And interestingly enough, again, like the East Bay sites are displaying some of those really high concentrations of nitrate, which could be a result of either septic waste or fertilizer runoff. But again, preliminary data, more will be coming, so stay tuned.
Just really quickly before we move on to Penny's presentation, can you guys hear me better this way?
Yes.
I can
hear you better.
Okay, we made a little technical change, so hopefully this will help a lot. So now we'll turn it over to, I guess before we do, does anyone have any questions for Anya about data?
Yes, I do, Ruth. Where did you say that this is Carletta, by the way. Where did you say that wastewater treatment plant was?
So that is it is right below the Mobile River site. It's orange there on sort of the northwest part of the map, and it has a little star on it. If you could point that out on that map, Brandy.
I think the cursor's too high.
Oh, is it okay? Then I'm seeing a different screen. I think I was trying to follow along.
Okay,
but
orange dot.
But that's that's just right outside of downtown Mobile Bay, or sorry, downtown Mobile.
Okay, thank you.
Yeah, Carletta, that is the major Mobile, the Clifton C. Williams wastewater treatment plant that's right there at the state docks.
Gotcha. Okay, thank you.
Now I can see the correct screen.
I have one question, Anya, because it's just a little hard to see on the screen. When you removed the wastewater treatment plant and then you were showing the other nutrients, what was that other site that popped up really high there with the dark green? That's mostly the, that's mostly.
Yes. That is the Fish River site. So that's just north of our Weeks Bay site.
Thank you very much.
Sure.
Now remember, you won't control the slides. So you have to...
All right, thank you. Turning it over to Penny, who has been doing some work on the molecular side with quantitative PCR and environmental or extracellular DNA.
Hi, I'm Penny, and like Brandy said, I'm working on the molecular part of this project. So I just wanted to give, my camera's on now, but I just wanted to give everyone an overview of what that entails. So we're using two different assays for the molecular side of this project. We're using qPCR or quantitative PCR to target specific sources of fecal contamination to Mobile Bay. And for this, we're targeting Bacteroidetes, which is a gut bacteria. So we have primers to target general Bacteroidetes to serve as like our total count of fecal associated contamination. And we have primers that are associated with humans, pigs, cows, and dogs, because those are all potential fecal sources to the area. And we are also using environmental DNA or eDNA metabarcoding to get an idea of everything that is in the water. With this assay, we will get DNA sequences from anything that has touched the water, passed by the water, lives in the water. We're going to get a ton of microbes, which is great. So yeah. And with this, we have 3 different sample types. So this figure is a lot. It shows a lot, but the main takeaway from this figure is that for the qPCR, we have 3 different sample types. So the EPA grant covers the water. And then, like Anya said, we were able to leverage oyster samples and sediment samples at a subset of the sites and on a seasonal timescale. And with the eDNA, we will be doing that for the water and the sediment samples. The oysters will not get eDNA. They will get a 16S rRNA microbiome, which will just show us the microbes that are in the oysters, because if we went and did an eDNA analysis on the oysters, we would just get oyster. But that is like the equivalent for the eDNA in the oysters. So we have 3 different sample types for all of the molecular analyses. And this is an overview of the processing that goes on. So at all of our sites, the right side of this figure, that's just the water filters. So we have different filters for the qPCR that we're doing in triplicate, and a different filter for the eDNA because we need a large volume of water for that. And then the left side of this figure shows those 3, the subset of sites that get the water, the sediment, and the oysters. And the first step for the eDNA analysis and the qPCR analysis is to extract all of the samples. So whether it's starting from the filter or the sediment sample or the oyster homogenate. And that is what I have been doing these past few months. So I started with the water filters and the sediment, and I was able to extract almost all of it. So the next slide, I think I put little, so I extracted water samples for qPCR from September 2022 to July 2023. So I just have 2 more months of water filters for qPCR to do. I extracted all of the water and sediment samples for eDNA. And I sent all of those eDNA samples for Illumina sequencing on October 9th. And we have already gotten some preliminary data back from that, which is very exciting. And I think we're next in the queue. They post a queue on the Integrated Microbiome Resource website. So we're close up in the queue for that, which is very exciting. And I'm back in the lab extracting those last few qPCR filters, as well as the oyster samples will be next after I finish those qPCR filters. Brandy, I think you're muted.
Yeah, I just realized that. Before, thank you, we move on, is there any questions for Penny's portion of it? She doesn't have yet the bioinformatics done for her data, but she's moving along at a nice pace.
Hi, this is Christina. I just had 2 quick questions. First one, just curious, did you consider adding birds to your sources of DNA? That's my first question. The second one, just kind of curious how you purify the DNA as well and made sure that it was, I guess, diverse and all representative, if that makes sense.
Yes, so to answer your first question, we have been working with multiple bird primers. We were trying to focus on a Canada goose primer because they're pretty prevalent in this area. And I tried 4 different primers and none of them worked. They either didn't amplify any of the DNA or they just amplified against all of our different poop samples. So I've been looking into either like a general avian primer or maybe like a seagull primer, but it's difficult because a lot of the other primers that are out there don't target Bacteroidales for the birds. They target a different species of bacteria. So we would have to think about if we target a different species, it won't go with our total or general Bacteroidales primers. So the birds are a little bit of a sticky point, but we're working on them and we do want to capture that foul source. For seeing if our DNA was pure, do you mean before we sent it for sequencing?
For validation, your qPCR validation.
Okay, yeah. So after I extracted all of my samples, I ran 16S I ran them with just like general 16S PCR and ran them on a gel to make sure that they amplified before I sent them for sequencing.
All right, thank you.
Can I ask a quick follow-up question? Was it the Grand Bay Bear Symposium where someone had used a bird?
Yeah, Amanda Free.
Okay.
Use the general avian primer.
Do they not work?
I haven't tried it, but it's not targeting Bacteroidales. Yeah.
So it targets a different bacteria species. So we need to have some conversations about whether we want to keep it all with Bacteroidales or include a separate, fifth, bacteria species. And that might be something that we can discuss with Amanda and, and others too, and Jessica Jones. That's a great question. Okay, we have a new team member that has been with us for a few months, but new to the stakeholders. This is her first stakeholder meeting. This is Brianna Jansen, who goes by Bree, and she's been working with us on the metadata clearinghouse. And so I'll turn that over to Bree and Ruth.
Hi, my name is Bree Jansen. I'm the new research technician for the project. I curate the entries that come to the clearinghouse, adding data that's related to microbial source tracking. So far, we have 29 entries within the clearinghouse, but we would absolutely love to have more, which is why we encourage the sharing of the questionnaire, which I'm sure some of you have seen pop up in your inbox, with community partners and anyone with research related to microbial source tracking. With the continued support of Pat David, who is a data architect over at the Dolphin Island Sea Lab, we have made significant improvements already to the website and will continue to add more. So the purpose of the clearinghouse itself is to compile all of this microbial source tracking data into one location so that it's easily accessible for anyone who would like to utilize it. This QR code does go to the wastewater footprint website, which is an awesome resource to use. The metadata clearinghouse is on that website. So the improvements that we've made, we have improved the links. Dr. Curtis, if you would click. Just go next. Or not. I guess it's not working. It's fine. Could you go back? I was hoping it worked. It wouldn't. It's okay. So we have improved the links, so we've made them clickable, and I've gone through and I've ensured that they all work. We've also added this new feature to search by column, whereas previously you can only use the search box that you had up there. Now you can go through and you can search by organization name, you can search by indicator that you would like, you can search by methodology, you can search by time period. So that is a new feature that we have added. And then I don't know why my things aren't working, but I did have a map of the focus area for the project on here. That was specifically created to showcase that we would like to focus on the Mobile Bay area. Yeah. So what I'm doing is I'm doing a analysis of the metadata to examine how policymakers, researchers, managers, and aquaculturists can better access and utilize this data. So far, I have done a temporal analysis, and I plan to do more of it specifically with indicators and their associated methodologies over time. I also further intend to do spatial and spatial temporal, so geographic location and geographic location over time. This is showcasing all of the metadata that we have within the clearinghouse. We have nutrient data dating back to 1953, and we have all other indicators that go from 1990 up until 2023. What I intend to do with this data is I intend to track the evolution of microbial source tracking data and investigate how the data is being utilized, whether it be for writing public articles or forming policies.
Before we move on to the next section, are there any questions about the clearinghouse? Or any suggestions too that that we can incorporate?
Yes.
And I also just want to give a pitch really quickly. I mean, first of all of our students are just absolutely amazing and have done such a great job. And so I really want to take a minute to recognize the hard work that they've done. We're so pleased that we've finished all of the field work, and now we can really dig into the samples and what they mean. But I also want to give an extra special shout out to Bree, because she came into this project most recently and has really gotten this clearinghouse, which was sort of a dream that we wanted to pull together all of these historical data to make them more available for other researchers, for students who might be wanting to access data, and also for, you know, all of you folks who are managers, who are folks in the industry, whatever it is, whether it's fisheries, aquaculture, you know, of conservation, and just make it easier for you to have access to these various indicator data. And Bree has just done a great job of jumping on board, getting up to speed, understanding this clearinghouse better than any of us do now, and actually making it a really useful tool. So for those of you who have been with us since the beginning, you saw this when it was just, you know, basically us begging for information about your data. And now you can see what that's already telling us. And this, you know, Bree's just giving you a little tiny teaser here of the cool things that she's going to do and that she's already been poking around with. But this is really exciting. And this is, I hope every one of you is as excited about this as we are, as you know, how this is all coming together. So thank you.
Absolutely. Thank you for that, that as well. And yes, Bree plans on writing an executive summary about, you know, where the data, as the data has evolved, and also the data gaps that exist, which hopefully is hypothesis driving in the future. So, you know, we hope to see future studies coming just out of this metadata analysis alone. Okay. All right, and I wanted to give you an update on the eDNA workshop that we are planning, and it will be a team effort. I'm just going to be voicing the update briefly. This is something that's, you know, of course, in the works, and would love to hear your input on what you would like to see from the eDNA toolkit. This will be an interactive, you know, half-day workshop. If there's, you know, sufficient interest to delve into some of these methods, we'd be happy to make it a full day workshop. So we're again still just designing it. We're anticipating this will be next fall at some point, fall 2024 at the Dauphin Island Sea Lab. We're going to go through the process of acquiring and analyzing environmental or extracellular DNA. This can be for new users, or it can be for those that you know, are an old hand at this. It will include kind of like a cooking demonstration, hands-on sequencing demonstration using a nanopore mini-ion sequencer that we have, and we can use for you know, screening samples and also small-scale projects. This particular project that we're doing right now with you know, total of 96 samples plus oysters and sediment. Of course, we sent it for sequencing on an Illumina sequencer because of the volume. But for small projects, the mini-ion sequencer will be great. I also walk you through you know, the bioinformatic analysis, which is it is very in a tractable ways, something that is for all users or all levels of users can work with, and it'll be free for the participants. So, but if there are things in particular that that you want to see incorporated into the workshop, you want to learn about, or you know, delve into further, we'd love to hear from you about that as we're developing this. And if you're interested in participating, we'll put out a call next summer for, you know, to sign up or apply to, you know, participate in the workshop. We've had the great opportunity to, again, leverage data collection and work with our other stakeholders as well. We've been able to leverage additional sample types, sample analyses, and then also sample sites, you know, working with, in particular, the Mobile Bay Keeper on the Dog River. And I'm going to turn this over to Penny to talk a little bit more about that.
Hi. So we partnered with Mobile Bay Keeper. I believe Cassie's on the call. Hi, Cassie. But we started this last fall, and we had a dry and a wet sampling event in the Dog River. We wanted to see the effect of a heavy rainfall event versus a dry period. And we went out to, I believe there's 9 sites that we did the qPCR at. I think there were a few others that they just did some bacterial analyses on. But we went out to these sites for those 2 events in 2022. And then we went out again this fall and we repeated that sampling. We again had a dry and a wet event. And we sent out, so we analyzed all of the data for last year's. We actually sent out the eDNA sample from 2022 with the rest of those eDNA samples that I sent out in early October. And if you go to the next slide, I have data. So this is the qPCR data from Dog River 2022. The dry condition is on the top and the wet condition is on the bottom. The scale for both of them is the same, just to show you the difference in the concentrations. There were some of the more upstream sites, which are on the left-hand side of the figure, had a lot higher concentrations of Bacteroidales during the wet event, which just shows that there's a lot of runoff into the Dog River. And the total Bacteroidales marker is that blue bar, and that was the highest for all of them. And out of our specific markers, the red, which is the human, was consistently the major source. It was a lot larger than the cow, the dog, or the hog for any of the sites. And then this is compared to the Enterococcus data that Mobile Bay Keeper generated. It's dry on the left for the qPCR on the top and the Enterococcus on the bottom, and then wet on the right with qPCR on the top and Enterococcus on the bottom. And you can see that they correlate really nicely. The scales for the enterococcus are not the same because there is such a large difference. The dry sampling event has a line that indicates EPA safe to swim thresholds. In the wet event, that line is actually like on the bottom. It looks like it's actually the axis because those numbers were so high for the wet event. But we saw that same trend in both the molecular analyses and the traditional analyses, which was really nice that we got to see that correlation. And we're working on the data that we just collected this year, so more to come.
All right, thank you, Penny. I appreciate it. Are there any questions, or Cassie, if you'd like to chime in, be glad to, and this is really, Penny's presenting it, but it's the, Cassie has been the, brainchild behind this.
Yeah, thank you, Penny, for going over all that and for presenting this recently at a poster session. Just wanted to share that I was actually fiddling with the data yesterday and today for this year, and it looks like so far there's a similar trend again with bacteria levels being significantly higher during the wet session versus the dry. And it looks like also some of the more upstream sites were higher again, just like they were last year, which is really something we're interested in Baykeeper, is not only understanding what the point source origins could be in terms of animal versus human input, but also which tributaries are contributing more compared to others, so we can potentially pinpoint those sources and work with partners to intervene on those pollution issues. So definitely excited about continuing to look at that data and getting qPCR data back for this year too. Thanks.
Thank you.
Awesome. All right, just to kind of wrap this up and moving forward, as we mentioned, that we got our dedicated field work has been completed as of the last sediment and oyster sampling happened in October, just last month. And so we're going to be doing more analyzing data, compiling, of course, the existing metadata, as Bree mentioned, and doing kind of a analysis, an executive summary of all of the source tracking related data. And I do want to, emphasize that, the data that you might be thinking about may not be, specifically a microbial indicator, but it could also be, nutrient data or, 16S microbiome data, or maybe it's, other things that, even isotope data that we can use as a, source tracking. So, don't, if you have data or you know about data or know something that's missing, it doesn't necessarily need to be Bacteroidetes specific, or, the other indicators that we mentioned, the Enterococcus or E. coli. There could be other ones that, you might think should be included. And if you have any doubts, please let us know, and we can help, sort that out, or and think about whether that kind of needs to be included into these analyses. And our clearinghouse is still continuing to be improved and updated in that way. We do plan to have another working group meeting, which is you guys, the stakeholder meeting, in about 6 months as well, consistent with this year, and to be on the lookout for announcements about the Eating A Toolkit, that workshop. And I think Ruth has some more information, perhaps on some more initiatives that are happening that she's spearheading at Dauphin Island Sea Lab that would be of relevant interest.
Yeah, thank you so much. Those of you who were with us before have heard me give my little elevator pitch about understanding those reciprocal links between the things that people do to our coastal systems, and then in turn how that can, affect us directly and indirectly. I think the big thing, particularly for this project, is just remembering that, ultimately our goal is to collect data that will help us create a healthier community. And certainly thinking about those links between ocean and coastal water health and overall ecosystem health and organismal health, and then how that translates to human health is really important. And obviously sometimes it's more direct, like many of the things that we're talking about, if we're putting, human sewage into coastal waters, that can be conveying bacteria and viruses that can make us sick. But also it's the other components as well. Is it affecting our seafood safety? And is it affecting our waters, and are they safe to swim in? And what is that going to do overall to the aesthetics as well of our environment? And that definitely affects our coastal economies by affecting commercial and recreational fisheries and our ecotourism. And so all of these things also, feedback on our own well-being and our own bottom line. And so constantly being, remembering that that's the kind of big picture question, and those impacts are what are ultimately driving this project and many of the others that we're working on here at the Dauphin Island Sea Lab and at the University of South Alabama. And so we're going to, continually be tying those components together as we continue to tell you about our progress on this project. And also just, keep that in mind. I encourage all of you to please do go visit our wastewater footprint. You can just Google that, our wastewater footprint. You can find our website. You can go on, you can listen to the replay of this presentation, see the data. Thank you for going to the next slide. That was perfect. And definitely give us feedback. I mean, that is intended to be a living resource. And, ultimately that's what we need to make sure that we are giving each of you the data that's of greatest use to you, but also in a way that is going to be the most useful to you. So, please explore that webpage. Please do, when we say we want feedback, we actually mean it. We really want that feedback. And, if you have data or you know of someone else who's collecting similar types of data and you can recommend them to the clearinghouse, I know Bree would love to talk to them. And also know, one of the big things that has come up a lot recently, and I'm so glad to see like members of the NEP, the CAC here, Shamika, I'm glad to see you here. Mimi, I'm glad to see you here. One of the things that came up in that meeting when we gave a presentation of this work was a little bit of disappointment that people feel like the data aren't being used. And, our job is to make sure that those data are available, accessible, shared, and used. And so that, partnering with this project, we hope will help address one of those issues as well. So I thank you all so much for joining us today, and hope you'll dig into our data and our resources.
Thank you. I'd like to open up for any other questions that might be out there.
This is Mimi Fern. I just had one. Looking at the map, it was really hard to tell. What are the 3 sites that you actually did the oysters and the sediment on?
Let's go back.
Yeah, that's a good question.
Here we go. Let's just pull up this map.
Yeah, I can answer that. Sorry, this map is so small, but basically, so the 3 sites were Foul River Bay. So that is FRB. That's sort of that pale light blue color on the bottom in the eastern Mississippi Sound on the left. And then we had Fish River, which is the sort of medium bright green on the East Bay, north of Weeks Bay, just north of that site. And then Dog River in the yellow on the East Bay was our 3rd site. And then we had control oysters as well.
Okay, is that the side that's right at the mouth of Dog River?
Yes.
Okay, thank you.
Yeah, we were planning on doing additional sites, but it's pretty labor intensive. And we did have some, it's the double-edged sword that we're trying to understand the inputs, freshwater inputs, but freshwater inputs also kill oysters.
And also not all of these sites have a sedimented bottom, so that did narrow down our choices. It's a great question. Any other questions?
So,
Well, this is
oh, sorry, I was just gonna say, Ted made a comment in the
thank
chat. Slides are no longer showing. I'm not sure if that's.
Oh, can everyone see the slides?
I see him.
Yeah.
It's just showing as a green bar.
Green bar. Oh,
I can see him just fine.
Yeah, it looks like me too. I just want to double check.
Apologize for that technical difficulty, Ted.
Well, this is Yu Chengfeng. I have a question about the sampling time for the dry weather data. That was, I guess, the project in collaboration with Mobile Bay Keeper. So for wet season samples, so how, I assume you went out after rainfall event. So how many days, how soon after the rainfall event did you guys collect the samples?
I can answer that question. So we were using a heavy rainfall marker of, I believe it was rainfall in exceedance of at least a quarter to half an inch of rain. And when we went post heavy rainfall in 2022 and this year, I think the accumulative rainfall at that point was closer to an inch or over an inch. And that time frame was, I believe, within 48 hours of sampling is when that rainfall had happened. And for the dry weather on both years, for both sampling sessions, we got lucky and we had I think a time period of at least 2 to 3 weeks where there wasn't rainfall. So we in 2022 we did dry first and then wet, which was great. And then this year it was the other way around. And I wish I could have been able to go out in the same manner of dry and wet again, but that was kind of our threshold for what we considered heavy rainfall to be.
Thank you.
Are there any other questions? And feel free to email us, or also if you think about anything in the future, please reach out. And also you will have access to this recording through the our wastewater footprint website. Well, thank you everyone for being here. We do appreciate it. We appreciate each of our stakeholders. All of you have played a role in, either developing this project or implementing it, and also growing it in the ways that we have. And so we appreciate everyone for being here and for taking your time to, to ask these questions and to also provide feedback. Thank you, everyone.